Now, second part of the applications of affinity chromatography, we'll see how it can be used in other fields. Affinity chromatography can be used in immobilized some purification and separation of immobilized metals and related compounds. It has used in biomedical analysis by biomedical fields. It is based on specific coordinate covalent bond of amino acids with metals. Metal ions that are immobilized within a column through the use of chelating agents, and it is developed for the isolation and purification of metal and histidine containing proteins. ज़ाहिर है मेटल containing proteins या अगर उसमें histidine containing जो proteins होती हैं, amino acid containing proteins होती हैं, ज़ाहिर है उनको हम isolate कर सकते हैं इस method से. Elution factors which are important during this type of affinitography include pH, ionic strength, this competitor, some competitor can also be used. So this is immobilized metal ion affinity chromatography. Now, analytical affinity chromatography, as the name indicates, it is used to study interactions in biological systems. It is also used to examine drug binding with agents that include serum proteins. They may have enzymes and some other receptors. Similarly, it is also possible to analyze the interaction of biomolecules with lectins, with aptamers, and with antibodies. These types of interactions of inhibitors with immobilized nicotinic acetylcholine receptors, they're also studied through this method. Now, biomedical affinity chromatography, this method or this approach may use affinity ligands that are substrates, inhibitors, cofactors, or some other types of components. They can also be separated. These are associated with biochemical pathways of target enzymes for screening potential enzyme inhibitors. ज़ाहिर है जो different metabolic pathways होते हैं, जैसे TCA cycle होता है, इसी तरह से और different glycolysis है और different metabolic pathways होते हैं, उनमें particular enzyme inhibitors को देखने के लिए या उनकी स्क्रीनिंग के लिए हम ये biomedical affinity chromatography जो यूज़ कर सकते हैं. It has also applications in the research in drug metabolism or the digestion of proteins for peptide mapping. So drug metabolism के लिए, digestion of proteins के लिए, या इसी तरह से और related इसकी applications बन जाती हैं. Denatured molecules may also be separated from the normal one using the same type of affinity chromatography. So these are different applications of biomedical affinity chromatography or affinity chromatography.